Hello, this is Professor Torsen. This is the recorded lecture for Chapter 12. In Chapter 12, we're going to learn how DNA replication occurs. I have 3 slides at the beginning of this lecture in which I summarize the history of DNA research. To keep this video from being too long, I will let you read about the history of DNA research on your own. Let's begin with this slide. We've looked at this slide several times already. I'm going to focus my attention on this end of the slide in which we are looking at DNA represented as the double helix structure here. The blue ribbon is representing the sugar phosphate backbone. And there are 2 blue ribbons because remember, DNA is double-stranded. There are 2 strands of DNA. Here in the middle are the colorful bars that are representing the nitrogenous bases and how they are hydrogen bonded to one another. So let's quickly review the structure of nucleotides. DNA and RNA are polymers. Polymers, we know, are composed of many monomer units that have been joined together. The monomer unit of DNA and RNA are nucleotides. Nucleotides are composed of 3 parts: the 5-carbon sugar, the phosphate functional group, and the nitrogenous base. Right? So we have the sugar in this purplish kind of color, the phosphate group in this yellow, and the nitrogenous base here in blue. So monomer units are combined through condensation reactions. We learned this in Chapter 3. The resulting bond that joins the 2 monomer units together after a condensation reaction has occurred is represented here in this dark red color right here. So this bond right here is called the phosphodiester bond. And this bond is what is joining this top nucleotide with this bottom nucleotide. Notice there is a water molecule represented because we are looking at the result of a condensation reaction, and we always lose 1 molecule of water in a condensation reaction. If we look at the structure of DNA and how those 2 strands line up with each other, this is a great figure. So we see individual nucleotides. Here, again, we have the 5-carbon sugar in this purple color, the phosphate functional group here in yellow, and the nitrogenous base here in green. And this long vertical bond here is the phosphodiester bond that is joining this nucleotide with the next nucleotide. Also notice how the complementary bases are paired up and hydrogen bonding with each other. So we've learned that adenine and thymine always pair together, and guanine and cytosine always pair together. Right, so all of these dotted lines are hydrogen bonds, and those hydrogen bonds are what are holding this left-hand strand and this right-hand strand together. Notice that these strands are anti-parallel. They are going in opposite directions. The 5' end of the left strand is up top and matched with the 3' end of the right strand. And at the bottom, again, the 5' end of one strand is matched up with the 3' end of the opposite strand. If you look more closely at the nucleotides, we see the nucleotides are also upside down from the left-hand strand. DNA replicates in a semi-conservative way. What this means is that we start with one parental molecule of DNA, as represented here in red. So this is my original copy of DNA. Notice it's representing 2 strands here, but we don't see the hydrogen bonds or the complementary base pairing. So we're representing DNA as these red strands here. So each one of these red strands represents the parental DNA. During DNA replication, those 2 parental strands separate from one another. And then the parental strands can be used as a template to make new strands. So we see that after one round of replication, we end up with 2 copies of DNA. And each copy is half old and half new. Each copy is one old parental strand here in red and one new daughter strand in blue. On this slide, we can practice coming up with the sequence of the complementary strand of DNA. So shown on this slide in black are 2 different parental strands of DNA. So this is one, and this on the right-hand side is a different one. And this would represent the template strand. So see if you can fill in the sequence of what the newly synthesized daughter strand, which remember is complementary to the parental strand, for this example and this example. So feel free to pause the video and work on that. Okay, let's see how we did. So on the left-hand side, I am looking at a single strand of DNA. This is our parental strand or template strand. Notice the 5' end is on the left and the 3' end is on the right. And the sequence of nucleotides can be seen here. So hopefully you remembered that strands are anti-parallel. They run in opposite directions. So if the 5' end of the parental strand is on the left, this needs to line up with the 3' end of the complementary strand. Then the bases should follow those base pairing rules. A pairs with T, T is pairing with A, good, and then G and C pair together, and then on this end I have the 3' up top and the 5' end at the bottom. Here is the answer for the second example. Right, I put the 3' end here on the left just to mix things up. So hopefully you put the 5' end of the new strand here on the bottom, and then follow those base pairing rules so that G and C always pair together and T and A always pair together. So DNA is very long. And so the question is, where should we start? Well, let's start at the origin of replication. Super original name, I know, but this origin of replication is a specific location on DNA where the 2 parental strands are separated, and then enzymes can come in and synthesize the new strand. So because DNA in eukaryotes is linear, It's one long strand. Our chromosomes will have many different origins of replication occurring throughout the length of the chromosome. But if we look at each individual origin of replication, it would be represented like either of these two figures here on the left-hand side. So let's look at the left-hand side. This is a more simplistic representation. What you see is the two parental strands of DNA here in black, and they are being separated by this green triangular shape here. This green shape that is separating the two strands of the parental DNA is the enzyme helicase. So helicase separates these two strands, essentially unwinding the DNA. A more detailed figure is shown on the right-hand side, but we are looking at the same thing. So we have the two parental strands of DNA shown in red, and here is the enzyme DNA helicase that is separating these two parental strands. These two parental strands here in red are surrounded by these sort of brownish orangish colors that look like shrimp or prawns to me. So these shrimp that are hanging on the parental strands are actually proteins. These are single-stranded binding proteins, and they help keep the two parental strands of DNA separated, because the two parental strands could easily bond right back together again if we did not prevent that with the use of these single-stranded binding proteins. My analogy is, if you consider two magnets from your fridge, if they get close enough, they will join together and you can pull them apart, but as long as those two magnets are close enough, they will join back together. And that is similar to these parental strands of DNA. If they are close enough together and not blocked by these single-stranded binding proteins, they would simply form those hydrogen bonds and be joined together again. One more enzyme is pictured in this figure on the right-hand side. This is the enzyme DNA topoisomerase. This enzyme is responsible for relieving any kinks, knots, or supercoiling that may happen in the DNA molecule as a result of the work of DNA helicase. So it is there to maintain the overall structural double helix form of the DNA molecule. DNA replication begins with the two strands being separated, as we saw on slide 10. Here on slide 11, we're looking at the next phase. Once the two strands of DNA are separated, as shown in these figures here, we can begin forming the new strand. The main enzyme that does this is DNA polymerase. So DNA polymerase will read the sequence of the parental strand and, following those same base pairing rules that you and I know, synthesize the new strand. DNA polymerase, however, can't start with nothing. It needs a little help to get started, something called a primer. A primer is a short strand of RNA, pictured here in yellow. Once there is a little bit of this RNA present, DNA polymerase can attach and continue this new strand of DNA. The enzyme that creates the primer is called DNA primase. If we look at DNA replication a little bit more closely, we can see the addition of a new nucleotide occurring in this figure. So in this figure, we have the template or parental strand here in red, and the new daughter strand is being synthesized here in blue. So we see this next nucleotide in the sequence being added to the new strand. Happens to be a cytosine. When the nucleotide comes in, it comes in with 3 phosphate functional groups. So this is similar to the ATP molecule we learned about in Unit 2. If you remember, ATP is the energy currency of our cells, and ATP can donate its energy by transferring a phosphate group. Here in DNA, it happens in a very similar way. Incoming nucleotides have 3 phosphate groups, 2 more phosphate groups than they need. So these 2 additional phosphate groups are providing the energy necessary for the reaction of condensation to occur. Remember, this is a condensation reaction. We are taking a monomer unit and adding it to another to become a polymer. So this figure shows really nicely what I mentioned earlier regarding multiple origins of replication. So again, eukaryotic chromosomes are linear. So we see they are these very long strands. So rather than start at one end or the other end or somewhere in the middle, we actually will have several origins of replication occurring on any given chromosome. And those origins of replication then move towards one another until we have completed the replication process. So this figure here is using dotted lines to represent the new strands and solid lines to represent the template or parental strands. In this figure, we are looking at DNA replication in one particular replication fork. So we see in red the parental strands or template strands, and we see the new strands being formed here in blue. On each one of these new strands, we see a little bit of yellow, and that yellow is representing the primer. Remember, DNA polymerase is the enzyme that is synthesizing the new strand of DNA, but it needs a little help to get started. It needs a little bit of primer to get started. So we see that for both the top parental strand and the bottom parental strand, there are new daughter strands being synthesized. We know that DNA is anti-parallel. So this top parental strand is the 3' end up top. That means it will match up with the 5' end of the newly synthesized daughter strand, which it is doing here. So we see primer here in yellow, and then DNA polymerase is synthesizing towards the left-hand side. Now, this arrow is specific because DNA polymerase can only add nucleotides to the 3' end. So it can only move in this left direction on this top daughter strand. Okay? So this is called the leading strand. And as the DNA polymerase makes this daughter strand longer and longer, this replication fork will continue opening towards the left-hand side. So not pictured here are those other enzymes, DNA helicase and DNA topoisomerase, helping to unwind this replication fork and have it go further towards the left-hand side. But if we look at what's happening on the bottom strand, well, the bottom strand has the 5' end towards the right-hand side. So again, DNA polymerase can only work in one direction. It can only add nucleotides to the 3' end, which means DNA polymerase is synthesizing this bottom daughter strand from left to right. This will become an issue because the DNA is opening up in the leftward direction. So as the top daughter strand is synthesized continually, this bottom strand will end up being synthesized in pieces. And we can see this represented in this figure here. We're looking still at DNA, and now we see the DNA is now unwinded even further than it was in the previous slide. So this new daughter strand here is even longer. We see the first strand that we saw in this previous slide here is right here. So as the DNA unwound in the leftward direction, the DNA polymerase was kind of stranded over here on the right-hand side. And there was all of this bottom template strand available. So what happens is that primase enzyme comes in and synthesizes a short strand of RNA, the primer, and then DNA polymerase can attach and synthesize the new strand up until it reaches this first portion. So these are called Okazaki fragments, and these Okazaki fragments are what are making up the lagging strand. So the leading strand is up top, and it's called the leading strand because it is synthesized in a continuous fashion from the right-hand side to the left-hand side in the 5' to 3' direction. But this bottom strand is synthesized in pieces called Okazaki fragments, and because of that, it is called the lagging strand. In fact, eventually, those Okazaki fragments must be joined together, and there is a special enzyme called DNA ligase that will join these Okazaki fragments together so that we get one continuous daughter strand on the bottom. This method of using the parental strands as template strands and synthesizing the new strands, the leading and the lagging strands, works very well until you get to the very end of the chromosomes. The leading strand functions just fine, but the issue is with the lagging strand. At the very end of the replication process, the enzymes are unable to copy this last little bit of the DNA molecule or chromosome. As a result, the chromosomes will get shorter and shorter with each round of replication. So you can imagine this figure here representing a chromosome, and each line going from top to bottom represents the chromosome as it might look round after round of DNA replication, which is round after round of cell division. Because remember, the cells are replicating their DNA every time they want to divide. So the way around this end replication problem is to add on these protective end caps to the chromosomes here in red. These red portions are telomeres. Telomeres are short, non-coding sequences of DNA. By non-coding, we mean it's not a gene, it doesn't code for any gene product, any protein. It's more or less gibberish. So this serves as a protection because if we lose a little bit of this telomeric DNA, it doesn't matter. We're not losing genes. Some cells that divide rapidly and many, many times over, have a special enzyme called DNA telomerase. DNA telomerase will lengthen the telomeres when it gets too short. So you can think of telomeres kind of like this sentence here, so that if I wanted to copy and paste this particular sentence and I accidentally didn't copy quite all of it, it wouldn't matter. You would still get a sense of that particular sentence. Now, I mentioned DNA helicase earlier. It is the enzyme that separates the two parental strands of DNA. And we've mentioned these single-stranded binding proteins that help keep these separated parental strands separated. Here is a better representation of topoisomerase. Topoisomerase will relieve any tension or supercoiling that may result as this DNA helicase unzips the two strands of DNA. You can think about this like when you are separating two strands of a rope. The rest of the rope may get tangled. And a nice way to remember this particular enzyme is this fun comic here. The topoisomerase is trying to make sure the DNA stays relaxed, stays in a double helix, and doesn't supercoil or knot up on itself. This final figure is a great overall view of the main steps of DNA replication. We start with helicase, that is unwinding the parental DNA. We make sure to include those single-stranded binding proteins that help keep the separated parental strands separate. We see the leading strand being synthesized by the enzyme DNA polymerase. And we see the lagging strand synthesized in these short fragments called Okazaki fragments. Thank you for watching this video. Now go wash your hands.